Dear students, myself Dr. P.K. Sharma, Assistant Professor, Department of Zoology, DUPG College Dehradun. Today I will discuss a very important topic that is called immunoglobulin or antibodies. First of all, I would like to introduce about the antibody. What is antibody? An antibody, also known as an immunoglobulin, is a large Y-shaped protein produced mainly by plasma cell, that is also called B cell. It is used by immune system to neutralize the pathogens such as pathogenic bacteria and viruses. The antibody recognizes a unique molecule of the pathogen that is called as antigen. The production of antibody is the main function of the humoral immune system. Now we come to the topic on history. The first use of the term antibody occurred reported by Paul Ehrlich. The study of antibodies began in 1890 when Kitasato Shibasaburo described antibody activity against diphtheria and tetanus toxin. In 1904, Almond Wright discovered opsonization. It means antibody help in the clumping of the bacteria without killing them. Now, the chemical structure of the antibody. Antibodies are specialized serum glycoproteins that are produced by B cell or B lymphocytes. These generally contain 80 to 96% polypeptides and 4 to 18% carbohydrate. Each molecule of antibodies has one basic unit or monomer of protein. It is formed of 4 polypeptide chains in 2 identical pairs. The large pair has 2 identical heavy chain or H chain, and a smaller pair has 2 identical light chain or L chain. Now the morphological structure. Each polypeptide chain of immunoglobulin has a constant and variable region. First, about the constant region. It is the carboxy terminal half of each polypeptide and is called C region. This part of H chains not associated with antibody combining site. It determines the physical properties and biological activities of antibody. This is the structure that you can see. This is the antigen binding site. One antibody has 2 antigen binding site. It means one antibody play an important role in the clumping of 2 antigens. It generally contains 2 chains. One is light chain and another is heavy chains. Upper part is called the variable region and lower part is called the constant region. You can see the structure. Now, variable region. It is the amino terminal half of both heavy and light chains and is called V region or antigen binding fragment, that is also called Fab. The amino acids of variable region are different in different antibodies. The variable region of H and L chains combine to form antigen combining site and is analogous to the active site of an enzyme. Each monomeric immunoglobulin molecule has 2 antigen combining sites represented as 2 arms of Y. Therefore, a single antibody combines with 2 antigen molecules. Now the different types and functions of antibodies. Based on the difference in the heavy chain, antibodies are classified into 5 types. First antibody is IgG. It is the major circulating antibody present in human serum. It comprises almost 75 to 80% of total immunoglobulin in normal human serum. Each molecule of IgG is a monomer with a molecular weight of 1,50,000 daltons. Based on antigenic differences in H chains, IgG are of 4 types: IgG1, IgG2, IgG3 and IgG4. Now the functions of IgG. IgG antibody participates in anaphylactic type of reactions. IgG provides long-term resistance to a disease as a product of memory B lymphocytes. Booster injection of vaccines raises the level of these antibodies. IgG of maternal antibodies cross placenta and provide the passive immunity to the fetus. This is the structure of different type of antibodies. You can see IgG, IgE, IgD, IgM and IgA. Now the next immunoglobulin, IgA. It is present in the mucous membrane of respiratory and gastrointestinal tract and in external body secretions such as tears, saliva, seminal fluid, urine and colostrum, that is also called the first milk. It is the second most abundant class, constituting about 10 to 13%. IgA occurs in two different forms. First is serum immunoglobulin, that is a monomeric with molecular weight about 1,60,000 daltons. Second is secretory immunoglobulin A, is a dimer with molecular weight. Next, functions of immunoglobulin A. Immunoglobulin A is secreted by plasma cells within mucous membrane at various locations. It can neutralize virus and inhibits attachment of bacteria to epithelial cells. IgA provides resistance to the respiratory organs and gastrointestinal tract. Next, immunoglobulin M. It is the largest antibody with a molecular weight 9 lakh daltons. It is the principal antibody of primary antibody response. When B lymphocytes are stimulated, this antibody appears first. It comprises about 10% of the IgS of normal human serum. Because of their large size, IgM don't cross placenta. Next, immunoglobulin D. IgD was first observed as myeloma protein in patients that was suffering from cancer, especially myeloma. It is the antibody with a molecular weight 1,80,000 daltons. Because of their large size, IgM don't cross placenta. It is found in traces about 1% in the serum and is rapidly catabolized. It is found on the surface of B lymphocytes in the placental cord, blood and on cells in certain lymphatic leukemia. Ig is found in minute concentration. The H chains of Ig is longer than those of IgG. Its Fc fragments binds to mast cells and basophil. This antibody is associated with allergic or hypersensitivity reaction and is more in the serum of person with antibody mediated allergy.